Okay, so let's look at this metabolic pathways for energy use, and we have looked at how the energy is transferred from carbohydrates, from lipids and proteins into ATP. That's the process of respiration, and we had aerobic respiration, anaerobic respiration, and fermentation. Then we looked at how we get the energy from sunlight into the carbohydrates. That was process photosynthesis. So now we're sort of looking at the idea of how this energy is going to be used to do work in the cells. And one of the first things to keep in mind is that every energy transfer is an inefficient process. That is, every time there is a transfer of energy, some of the energy is going to be lost as heat, and that heat is not going to be usable by the cell. That heat is not going to be able to be retained by the cell. The heat is just going to dissipate from the cell, and so it's not going to be used to do work in the cell. The part that is going to be used to do work is where that phosphate group, so remember triphosphate, you've got 1, 2, 3 phosphates there, and that third phosphate is going to move over onto a protein, and it will attach to that protein, change the shape of the protein, allow the protein to do some type of work, and then that phosphate will be released. And so that, as energy transfers go, that is a very efficient form of energy transfer. Now, there is still a loss of some of that energy in the form of heat, but in general, biological energy transfers are more efficient than mechanical energy transfers. Okay, so let's go on down here and take a look. One of the things your textbook points out is that most of the ATP energy in a cell is used to build new cellular components, and basically prokaryotic cells tend to be a little better at this than eukaryotic cells. We might look at it as the idea that there's fewer cellular components in those prokaryotic cells to build. So prokaryotic cells can have a very rapid rate of reproduction, a very rapid turnover rate. And so, we've got all these different organelles in a human cell, plant cell, fungal cell. You don't have those organelles to build in a prokaryotic cell. So these prokaryotic cells can reproduce themselves very rapidly. Okay, so let's go on down here and take a look. Now here's one, the idea of using some of these biochemical tests to try to identify different species of mycobacteria. Remember, we've got this mycobacteria down there at the bottom left, tuberculosis. In other words, the mycobacterium bacteria that causes tuberculosis. Okay, so to be able to separate that from other mycobacteria, notice up there at the top you have the acid-fast test. Well, some of these acid-fast mycobacteria are going to be slow-growing mycobacteria. Some are going to be rapid-growing, and separate them out that way. Okay, among those that are slow-growing, we can use a urease test and see if it breaks down urea. If it doesn't, it's Mycobacterium avium. If it does, we can use a nitrate reductase test, and if it can reduce the nitrates, then it's the tuberculosis forming, it's the Mycobacterium tuberculosis. If not, it's the Mycobacterium bovis. So the main point here is that we can use this biochemical test to identify these different species of the Mycobacterium. Here's another biochemical test right down here. urease test, and basically if it hydrolyzes the urea, it'll produce ammonia. Ammonia is going to raise the pH, and again in this test it's using the pH indicator. So if it turns a purple color, there was an increase in pH. Okay, we had the term autotrophs earlier. Autotrophs can build their own organic molecules. This is still a process that requires ATP. So the sunlight the energy from sunlight is going to be used to make the ATP, and it's going to be used to split the water, and that hydrogen from the water is picked up on the NADP, forming NADPH. And so to do work in the cell, this idea of taking that carbon dioxide, attaching the hydrogens to it, making a larger, more complex molecule like a carbohydrate, it is going to take an input of energy. And yes, the plants, the photosynthetic bacteria, use sunlight as their initial source of energy, but that sunlight is converted to energy in ATP, and ATP is the one and only compound that can provide the energy to directly run work that is done in the cell. Okay. Notice the other line there in your textbook, heterotrophs. Heterotrophs, by contrast, have to have some source of organic compounds in order to carry out biosynthesis. Now, biosynthesis, if you synthesize something, you are making something. And of course, bio refers to a living organism. So, in other words, for that plant, for that photosynthetic bacteria to be able to make organic molecules, it has to take sunlight, convert that sunlight into energy that is contained in ATP and energy associated with the this hydrogen carrier, NADPH. In our case, we have to have some kind of organic molecule, such as glucose, that is energy rich, and then we're going to use the energy from that to make ATP and then to do cellular work. Okay, so in our case, in the case of heterotrophic bacteria, the cells are going to use these organic compounds both as a carbon source and as the energy source. Okay. And anytime you have a buildup of complexity of molecules, it's going to require an input of energy. Generally, in nature, you do not get a buildup of complexity happening on its own. Okay, so polysaccharide biosynthesis, in other words, making larger, more complex organic carbohydrates right there. Notice what we have right there. We've got process of glycolysis, and it's kind of this idea that the catabolic process feeds into the anabolic process. So the components of this breakdown process, glucose 6-phosphate, are going to feed into the process of making glycogen in both bacteria and animals, and we're going to need ATP to do this. Now, down there in the lower part of that diagram, you notice it says UTP. Okay, the A up there in ATP is the adenine. That is the same base that we have in DNA. So this ATP is a nucleotide. It consists of adenine, ribose sugar, which makes the adenosine, but adenine, ribose sugar, and then 3 phosphates. Well, down there at the bottom, we have another base, one that we have in RNA, uracil. Uracil, ribose sugar, and then 3 phosphates. Well, notice in the diagram, it's in that explosion symbol. So it's going to work just like the ATP. The uracil triphosphate is going to be an energy-rich compound, and it can transfer energy in there to link the smaller units to make those larger units that you see over on the right-hand side, the glycogen or the peptidoglycan. Okay, so let's go on down. If you go back to Krebs cycle, remember we had one in there called GTP and GDP, guanine triphosphate, guanine diphosphate. Again, that guanine is one of the bases that we have in DNA and RNA. It's one of the bases that's part of these nucleotides. And guess what? It worked just like these two. The guanine triphosphate was a higher energy molecule and an unstable molecule, and it was able to transfer the energy for doing work in the cell. Okay, so let's go on down here. Lipid biosynthesis. One of the things that they point out is that there's a lot of variation in lipids, so they're synthesized by a variety of routes. And right here, biosynthesis of lipids. From the middle of glycolysis there, glyceraldehyde phosphate, we're going to make glycerol. And then from the acetyl groups right there, we're going to make the fatty acids. If you recall, in the respiration of fats, this is where the glycerol and the fatty acids fed into the process of respiration, was in the middle of glycolysis as glyceraldehyde phosphate and as the acetyl coenzyme A at the beginning of Krebs cycle. Okay? Most of the lipids are going to wind up as these phospholipids, which are going to serve as cell membranes. Okay? So, things to keep in mind. And they talk about some other lipids there, like cholesterol and waxes and so forth. Waxes, that's a lipid that we find as a component in the acid-fast bacteria, such as the mycobacteria. Then also some of these different pigments, like the carotenoids and so forth, those are also lipids. Let's go down here to amino acids and proteins. In order to make proteins, you have to have amino acids. Okay, some microorganisms, like the Escherichia coli, are going to have the enzymes to use materials like glucose and inorganic salts to make these amino acids. And so they can make all the amino acids necessary that they need from the various intermediates of carbohydrate metabolism. Now, that is something that we cannot do. Okay? That we, being humans, animals, we cannot do that. We have to obtain intact amino acids from other organisms. So, basically, just like the idea of the food chain, the production of proteins is going to move from the bottom up. We have bacteria that can actually produce the amino acids from, we might say, from scratch. Right here, from taking carbohydrates or the intermediates in this process of respiration and changing those over to amino acids, combining those with the nitrogen groups, sulfur and so forth, and making the amino acids, something that we can't do. Plants are also able to do that. Okay, so right there, biosynthesis of amino acids. And notice here's where one of those alternative pathways comes into play, pentose phosphate pathway, and then the Entner-Doudoroff, you can read it right there in front of you. That other pathway comes into play. Okay, recall that those are two pathways, metabolic pathways, that we do not have. Those are pathways of respiration that eukaryotic organisms do not have. Okay? Now then, there's also the possibility of using the Krebs cycle right there as a means of producing these amino acids. So let's go on down here and look at this. We have what's called transamination. Okay, so what that amounts to is we can take the amino group that said nitrogen and 2 hydrogens, that group, that's the amino group. We can take the amino group off of one amino acid and put it onto a different organic acid and change that into an amino acid. Okay, so that is transamination. Okay, so transamination, the transfer of an amine group, okay, and deamination was the removal of an amine group, okay, of that nitrogen group. Okay, so we've got the terms. We had the term earlier of deamination, removal of that amino group. We have amination, the adding on of an amino group, and then transamination to remove the amino group from one amino acid, put it onto another. Most of the amino acids are going to be used to build proteins in cell, and again, in the building up process is going to require energy. That energy has to come from ATP. Okay, purines and pyrimidines, these are going to be part of the DNA and RNA. These are made up of units called nucleotides. Okay, these nucleotides consist of either a purine or pyrimidine, depending on what type of nitrogen-containing base is a part of that nucleotide, a 5-carbon sugar called a pentose, and a phosphate group. Okay? So, excuse me. And so goes through the process here of how this is made and discusses that. And let's go down here and look at the diagram and notice again what's going to happen is we're going to have intermediates from process of glycolysis that are going to be involved. We're going to have intermediates from the Krebs cycle that are going to be involved here. And then it's going to go through a series of metabolic pathways to wind up producing these nucleotides. Again, anytime you have a buildup of complexity, it is going to require an input of energy from ATP. Okay, so let's go look at this last part, integration of metabolism. And so it's the idea that we've looked at processes that microorganisms are going to use with energy from light, inorganic compounds, organic compounds, and to use this for biosynthesis, that is, to make new organic compounds. Now, we've looked at anabolic and catabolic reactions, that how these two work together, and that the intermediates from catabolic reaction, breaking down process, will be used in the anabolic process. Also, the energy derived from the catabolic process is used to drive the anabolic process. We've got this term amphibolic pathways. In other words, ampha means multiple, and multiple or dual. And so these metabolic pathways that can function in both a building up process and a breaking down process are called amphibolic pathways. As you see right there, it means dual purpose. And right here is a diagram in your textbook that just kind of shows some of the interactions and complexities of these pathways. Notice central to this is glycolysis and the Krebs cycle. Then you have all these branches going off of there for the amino acids, lactic acid, and so forth, various carbohydrates, and so forth. These amphibolic pathways are going to tie together the synthesis of carbohydrates, lipids, proteins, nucleotides, and so basically you can simultaneously have the breakdown of products while you at the same time have buildup of products taking place. Okay, and so let's see what we have right here. And then, of course, these things do not occur randomly. We have control mechanisms in this, and so we have some examples there of your control mechanisms. And then here's that term again, feedback inhibition. Feedback inhibition is a just fantastic mechanism that we see in all organisms, that as the end product begins to accumulate, feedback inhibition shuts down the metabolic pathway that's making that end product. And they give the example there with ATP. If the ATP starts to accumulate, then the feedback inhibition shuts down the process of glycolysis. Okay, that finally gets us to the end of chapter 5.